The traditional model of healthy eating has long centered on calorie counting, portion control, and macronutrient balance, principles that, while once necessary to curb overconsumption and manage weight, now seem increasingly inadequate in addressing the complex biological realities of aging. As science deepens our understanding of cellular health, it's clear that longevity and vitality depend on more than just managing weight or eating low-fat meals. Aging is fundamentally a process of accumulating cellular damage, metabolic rigidity, and chronic inflammation. And in that context, the future of healthy eating must shift from a surface-level focus on calories to a deeper, biologically integrated strategy rooted in autophagy activation, hormonal balance, and micronutrient optimization. Autophagy, often described as the body's internal recycling system, plays a central role in maintaining cellular integrity. It identifies and removes dysfunctional proteins, damaged organelles, and invading pathogens, essentially cleaning out what no longer serves the cell's function. This process is especially crucial as we age, because autophagic activity declines with time, contributing to the onset of age-related diseases like neurodegeneration, metabolic dysfunction, and cancer. What we eat, and when we eat, can either suppress or stimulate this process. High-calorie, high-carb, and high-frequency eating suppresses autophagy by constantly triggering insulin and mTOR, which signal the body to grow rather than repair. In contrast, metabolic states associated with fasting, carbohydrate restriction, or nutrient stress, like polyphenol or AMPK-activating compounds, upregulate autophagy, allowing cells to restore balance. Thus, healthy eating for aging should no longer be defined by how little we eat, but by how wisely we eat to promote cycles of repair. This doesn't necessarily mean extreme caloric restriction or perpetual fasting, but rather creating dietary patterns that cyclically enable autophagic activation without nutrient deprivation. For example, eating within time-restricted windows, practicing periodic low-protein or fasting-mimicking meals, and avoiding constant snacking can all serve to mimic the evolutionary stresses that once kept our cellular repair systems in balance. Yet, autophagy alone is not enough. The hormonal and nutrient signaling systems that orchestrate metabolism must also be stabilized. In aging, hormonal regulation becomes increasingly fragile. Insulin sensitivity declines, cortisol rises, thyroid activity slows, and sex hormones fluctuate. Standard dietary advice, such as frequent low-fat meals or diets high in refined grains, can worsen these imbalances by promoting insulin spikes, blood sugar instability, and chronic inflammation. Instead, diets that support stable blood sugar via lower glycemic load, encourage metabolic flexibility via balanced fat and protein, and reduce inflammatory load via nutrient-dense, anti-inflammatory foods, can rebalance hormonal systems that directly affect energy, cognition, and resilience. Micronutrient density, long a secondary concern in popular diet culture, must also take center stage in aging nutrition. Subclinical deficiencies in vitamins and minerals are far more common than most realize, especially in older adults with diminished absorption or restricted diets. Vitamins A, D, K2, C, E, and the B-complex, along with key minerals like magnesium, selenium, and zinc, are not merely supportive. They are actively involved in cellular maintenance, DNA repair, redox balance, and autophagy. Their absence, even in marginal amounts, can impair mitochondrial function, slow detoxification, and accelerate biological aging. A calorie-restricted but nutrient-poor diet, often mistakenly labeled healthy, may do more harm than good. Equally important is how these micronutrients are delivered. Whole foods remain the gold standard, as they contain synergistic compounds that work in harmony, such as polyphenol, fibers, and cofactors. Foods like leafy greens, wild-caught fish, organ meats, fermented vegetables, seaweed, nuts, and berries are vastly more protective against aging than low-fat dairy or fortified cereals. This is not simply a matter of natural versus processed. It is about the density, bioavailability, and functionality of what we consume. What we're witnessing is the slow collapse of outdated diet paradigms. Calorie counting and macronutrient math don't address mitochondrial dysfunction, oxidative stress, or cellular senescence, the actual drivers of aging. A new model of healthy eating must instead prioritize cellular nutrition, food that not only fuels the body, but also clears waste, optimizes signals, and maintains integrity over time. It must account for the cyclical nature of metabolism, the need for periodic autophagic activation, and the non-negotiable requirement for micronutrients efficiency. In practice, this model looks like high-variety, whole-food eating patterns that incorporate deliberate metabolic stress, fasting, low glycemic days, while ensuring micronutrient abundance through ancestral foods. It involves replacing the treadmill of constant eating with timed meals that restore circadian alignment. It favors nutrient intelligence over calorie obsession and metabolic resilience over body size. And critically, it adapts to the changing biology of aging, recognizing that what worked at May 30th be harmful by 60. In conclusion, redefining healthy eating for aging is not about restricting more, it's about understanding more. It's about nourishing the body not just to prevent disease, but to sustain repair. As the science of longevity continues to advance, so too must our dietary frameworks. A calorie may still be a calorie in physics, but in biology, context is everything, and aging cells demand more than just fuel. They require precision, support, and intelligent design.